Welcome everyone tonight. We have our presenter tonight is Dr. Michael Levy. He is a bioinformatician. He did his undergrad in genetics, and then he has a PhD in biochemistry and developmental biology, and works on projects related to brain development, genetics, bioinformatics, and he's from Ontario, Canada, and works under the directorship of Dr. Bekim Sadikovic. Welcome, Mike, and thank you for presenting.
Great, thank you for inviting me. I'm very excited to talk about our work today. I'm coming to you from London, Ontario, Canada. I work at the Verspeten Clinical Genome Center at London Health Sciences Center. And today I'm going to be talking about DNA methylation episignatures, what they are, and how we are using them to help diagnose Mendelian neurodevelopmental disorders. And for those who don't know, a Mendelian disorder just means one that is caused by a single gene, as opposed to something like maybe autism, which can have a very complex underlying genetic cause. So I understand we may have a mixed audience, so I'll just start off with some brief background to kind of get everybody up to speed. So the central dogma of genetics is that we have our genes in the DNA, and it's transcribed into RNA. The RNA is translated into polypeptides, which make up proteins, and that's basically what your tissues and cells are made of. And then those come together to create what's called your phenotype, or your set of observable characteristics or traits of an organism. Now, when something goes wrong with the DNA sequences, when we have what's called either a mutation or a pathogenic variant, it means that this change is propagated to the RNA, to the proteins, and ultimately to the phenotype itself. And since one gene can have a role in many tissues of your body and many different cell types, a change in one gene can have effects throughout your whole body. And that's how we end up with syndromes, which have systemic effects throughout the body. Now, changes in the DNA sequence itself are not the only way that we can end up with such changes. Epigenetics are mechanisms that control gene activity without changing the underlying DNA sequence. So to explain what that is, if you look at your DNA in your cells organized into 23 pairs of chromosomes in human cells, and if you were to zoom in on that, you see that the DNA, it doesn't just float around by itself in the cells, it's wrapped around proteins called histones into a structure that's called nucleosome. And chemical changes to these structures can change gene expression. So for example, on these histone molecules, we can add chemical groups, acetyl groups, methyl groups, or phosphate groups to them, or we can add chemical changes to the DNA itself. And DNA, as you probably know, made up of A, T, Gs, and Cs. These methyl groups can be added onto the cytosine nucleotides. In particular, they tend to be added where cytosine nucleotides are directly followed by a guanine. And so we call these CpG sites. The P represents the phosphate backbone of the DNA. And depending on where these epigenetic modifications occur and exactly what the changes are, they can either turn a gene on or activate the gene, or they can turn a gene off or repress the gene. So for example, if we have acetylation added to the histones, or if we remove DNA methylation as shown by the white circles here, then the what's called chromatin will actually open up and the genes can turn on. And then the opposite, if we remove the acetylation and we add DNA methylation, then the chromatin will condense and the genes will turn off. And in this way, epigenetic changes can regulate gene expression. So these changes are usually heritable as cells divide. So if you have a change at early stages in development, then this will be tend to be propagated to different cell types, and ultimately you can see some of these changes in the fully differentiated cell types as well. Of course, not all of these changes can be propagated, and the differences in epigenetics and different cell types are what leads to this differentiation. The causes of these epigenetic changes, they can be caused by underlying changes in the DNA sequence. So there are genes that their function is to, for example, add DNA methylation or remove DNA methylation. And if you have a gene that's supposed to add DNA methylation and you have mutations, so it's not working properly, then you're not going to be adding it where you're supposed to. It's also environmentally responsive. So there's studies that have shown that epigenetic changes, for example, in response to a fetal alcohol syndrome, and in adults, even diet and exercise can play roles in epigenetic changes. It's generally plastic and reversible and changeable. Changes occur as a normal part of development. We know there are changes as we age. But while this is what happens normally during development, what we have been studying are what happens when there is a change in the underlying DNA sequence. So for example, if you were to have a change, a mutation in the DNA at a certain point in development, then these changes are going to be propagated down to the cells and the tissues that develop from them. And the changes in epigenetics will also get propagated as well. If you have a change in the earliest stages in development, like in the embryonic stem cells, that's going to affect all the tissues. And then when you have the fully differentiated tissues of the organism, we can see changes in epigenetics here that are reflections of what happened at the earliest stages in development. What this means is that we can use tissue, for example, blood cells, to study changes that occurred at the earliest stage of development in cells such as stem cells. And so that's what we do. We use blood samples to study their epigenetics to look for what we call an episignature. And an episignature is a recurring epigenetic pattern that's associated with a genetic or environmental etiology or cause in a particular patient population. So for example, if we take an unaffected individual, we can look at their DNA and we can look at the DNA methylation, and each one of these little lollipops here would represent one CpG, so one location where you could either have DNA methylation, as shown with the darker circles, or you could have no or lower DNA methylation as shown in the lighter colored circles. And you have an affected individual, either they have a syndrome or some type of environmental exposure, and they will have some places where their DNA methylation is unaffected and it's the same, and then you have other places where they have changes in their DNA methylation. And the combination of all of these places where they have changes are what we call the episignature, because it's like the signature that can be used to identify a particular syndrome. So to identify these episignatures, the first step is to actually find the patient. So most of the syndromes that we study are very rare, with only a few hundred to a few thousand patients in the whole world who have identified them. So we work with labs in many different countries to identify these patients. We take a blood sample and we isolate DNA from the blood. The DNA methylation is then measured throughout their genome. And then the results from those patients are compared with our database of other samples to find where these patients have differences. So the measuring of the DNA methylation is done using something called a microarray. In particular, we use one from a company called Illumina. It's called the Infinium Methylation Epic Microarray, or Epic Array for short. It's their newest version from their older versions, which were the 27K or 27,000 and the 450K arrays. The new versions, they measure CpGs at about 800, or they measure DNA methylation at about 850,000 CpG sites across the genome. Now there's about 30 million total in the genome, so we're only measuring a small fraction, but these microarrays are designed so that they target all the most important regions. So for example, we measure at 99% of all known genes and about 98% of CpG islands, and CpG islands are regions where we have many CpGs that are close to each other, and these tend to be areas that can regulate gene expression. But they also measure some areas outside CpG islands. Now these microarrays are actually pretty small. The actual size of this are about a few inches long, and each one of these little sectors here is for one sample, so we can measure 8 samples at a time on this. And each CpG is detected on the microwave with what's called a probe, and so you'll hear me refer to these as probes during the analysis. So we have identified several Cat6 related episignatures. We have a signature for the ARTHS syndrome caused by variants in Cat6A on chromosome 8. We've looked at this in 18 patients. For CAT6B on chromosome 10, we've looked at 4 patients who have GTPTS and 9 patients who have SBBYSS. The 2 syndromes caused by CAT6B, this is from a recent paper that looked at where the variants were located, and they showed that whether a patient has GTPTS or SBBYSS generally depends on where in the gene the variant occurs. So the CAT6B signatures for GTPTS and SBBYS, we actually had published those first a few years ago. So in 2018, we published a paper that had 9 of these signatures, including GTPTS and SBBYSS. In 2020, we published another paper with 34 signatures, including these 2. And then most recently, it's actually just coming out tomorrow, I believe, is the official publication date. Our newest paper has 57 of these episignatures, and to these, we've also added ARTHS. So since ARTHS is one of the new ones, I'm going to take you through the process of identifying that signature specifically. And so we basically use the same process for all these different episignatures. So the first step is to choose the samples that we use that will be what we call our case and our control samples. So for ARTHS, our case samples are going to be our 18 ARTHS samples, and the control samples are going to be 54 samples chosen randomly from our database. And 54, because this is a 3-to-1 ratio. So for each ARTHS sample, we chose 3 controls. They're random, but they're also matched for age and sex so that we have a similar proportion of males and females and young and old patients in the cases and controls. Now, for most episignatures, we would just leave it like that. That's we'd choose the controls. But because ARTHS caused by CAT6A is so similar to the CAT6B syndromes, we also included samples from the CAT6B patients. And this will let us more precisely differentiate between ARTHS and the other syndromes, as I'll show you on some subsequent slides. So using these samples, we want to find the most differentiating probe. So the places in the genome where the DNA methylation is the most difference between the cases and controls. And we use a number of different statistical and analytical steps to do this. So first we look for places where there's a high methylation difference, somewhere where the ARTHS samples have high methylation and the controls have low, or vice versa. We look for probes that have low p-values. That means that they are statistically significant. We use a statistical method called the receiver's operating characteristic curve, and this helps us balance true positive and false positive rates. We also remove probes that are highly correlated so that the probes that we do select have more unique values. And lastly, we remove highly variable probes. So we want probes that will act consistently, within the HRH samples and ARTHS samples, like consistently within the controls, so that we know they're going to be more reliable markers. So this is what's called a volcano plot. And on this plot, each one of these circles represents a single one of those 850,000 probes. And the ones in red are the ones that actually fulfilled our criteria that we selected as part of this episignature. On the x-axis down here, it's the methylation levels. So to the right are probes that had higher methylation in ARTHS, and on the left are ones that have low. And on the y-axis is the negative log of the p-value. And it's a negative, that means the higher the value here, the lower the p-value, or the more statistically significant the results are. So we can use those 115 probes, and we can do what's called unsupervised clustering with our 85 samples of the cases and the controls. And this is called hierarchical clustering. And what it does is this is an algorithm. We don't tell it whether the samples are ARTHS or controls. It looks at the DNA methylation, and samples that are more similar, it groups them together. And more different, it puts them farther apart. And we can see that all the ARTHS samples are more similar to each other, and they separate from the controls. And so using this way, we can identify which of the 2 clusters a sample is from. And this section here is called a heat map. And the colors represent the levels of methylation. So if it's red, it has high methylation, and if it's blue, it has low methylation. And you can see there's kind of like this dividing line here where we have slightly different patterns between the cases and the controls. We can use another method of unsupervised clustering. This is called multidimensional scaling, similar to the previous slide. in the previous method, where samples that are more similar to each other are closer together and samples that are more different are farther apart. But this looks at it in 2 dimensions, so we can get a better idea of the overall data structure. And I mentioned how it was important that we had to include the Cat6b samples as controls. And this shows why that is. So on the left, this shows what the result would look like if we didn't include them. So we can see how the ARTHS samples in red, they're nicely separated from the controls. But if we didn't have these colors at the top here, we wouldn't know if these samples are the ARTHS, SBBYSS, or GTPTS, because they're all kind of mixed together. But once we included these as controls, we now have this clear dividing line, and we can differentiate the syndromes from each other. On these multidimensional scaling plots as well, when we didn't include them, we can see that there's overlap between the ARTHS and the other samples. But once we include them as controls, then we have kind of these 3 separate clusters. We also use methods of supervised clustering as opposed to the unsupervised. So this is something that's called a support vector machine. It's a machine learning method to generate what's called a classifier. So a classifier is kind of like a mathematical model or a mathematical equation that can identify differences between 2 sets of data. So we give it a bunch of samples and we label them as either ARTHS as cases or as controls. That's why it's called supervised, because we're telling it what's the case and what's control. And the algorithm will look for the differences between the 2 groups. And then we give it a new set of samples, the testing set in gray that it has not seen before, and it will use the algorithm that it develops to classify these with a probability score of being they're probably an ARTHS sample or they're probably not an ARTHS sample. And on the left here is what we call an MVP, or methylation variant pathogenicity score. And a score that is close to 1 means that the sample has a high probability of being from a patient with ARTHS, and if it's close to 0, it means it has a low probability. And we tested this with samples from many other patients with many different syndromes. So on the bottom here are samples from patients with 1 of 56 other syndromes. And we can see that generally they have very low scores. So that shows that this classifier does a good job of finding the ARTHS samples and kind of ignoring the non-ARTHS samples. And you might see a few samples that have somewhat higher scores, so it's pretty common. These are never perfect classification methods, and so we will always investigate these further. So for example, if we can take this sample here and this sample here, we can go back to the unsupervised clustering, and we can see that those 2 samples, they are clearly not ARTHS samples. So we never use just the support vector machine classifier or just the unsupervised learning methods. We always use them in conjunction when we use these in the clinic to try to diagnose patients. So I mentioned how our previous papers found 38 identified signatures, and our most recent paper added 19 to this. So this method that I just described, we basically have gone through this method for every one of these signatures. And we can also look at these in comparison to each other. So each on this plot here along the bottom, each column here is a different signature. The arrows are showing the Cat6 signatures. And on the left is the methylation difference. And each circle is a probe from the episignature. So if it's up high, it means it's a probe that has higher methylation in whatever the syndrome is. And if it's low, it has lower methylation. You can see there are some syndromes that tend to have very low levels of methylation in the affected individuals, and some that tend to have higher methylation. And the Cat6 syndromes are generally in the middle, showing they have a balance of increased and decreased methylation. We made classifiers for all 57 of these episignatures. This is showing the classifiers along the bottom and the MVP probability scores along the side. And so for each column here, even though it's kind of hard to tell, but one column here, there's actually thousands of samples plotted here. It's just they mostly overlap. So for each column in red, we have the patients with that syndrome. And in black, we have every other patient, every other sample that we've tested. And we can see that the patient samples generally have very high scores, and the non-patient samples generally have very low scores. and we apply a method similar to this in a clinic, so when we get a sample, we will test that sample with every one of the classifiers to see which samples it has a high score for. And if it's a sample from one of our patients, then it'll have a high score for, from one of these signatures. Now, some of these ones that have more moderate ones, in our recent publication, we go into all of these in a lot of detail for anybody who's interested, but I won't go through all that now. I will mention one interesting set, which is for these syndromes here. It's a pair of syndromes called RSTS1 and RSTS2. And similar to the CAT6 syndromes, these two are very similar to each other. And so that's why we see that these blue ones are showing some of the samples that have kind of where we see overlap between these samples. And so we actually do have, we don't get a perfect separation for these ones. So what we're working on now, up until now, we've mostly been focusing on identifying these episignatures kind of one at a time and their clinical application. But now we've started to investigate some of the functional biology that could be underlying these. So for example, comparing signatures to each other to see which ones are more similar. That's what this plot over here is showing. We are trying to find genes that are, maybe there are genes that are affected in many of the signatures. It could mean that there are genes that are fundamental to neurodevelopment versus genes that are only affected in some signatures. Are there groups of syndromes that are more similar to each other? So that's the kind of thing that we're investigating now. In addition, we are, of course, always looking to expand our number of episignatures that we have. So we started publishing this work about 3 years ago. And for the last 3 years so far, we've had a new version of what we call our episign test and added more syndromes that we can diagnose using this method. We're currently working on our newest version, which is version 4, which will be released and available this year. And using this one, we expect that we'll be able to diagnose somewhere around 100 different episignatures, but that work is still ongoing. I'd just like to end by acknowledging our lab director, Dr. Beckham Sadikovic, who really started and kind of leads this work. Haley McConkey and Jennifer Kirchhoff from our lab, who I worked very closely with on this current project. The other members of our data analysis and bioinformatics team. And of course, we have a very large clinical lab with a few dozen people who do all the lab work for me. So I'm very thankful to all of them. So thank you for your time, and I'm happy to take any questions.